Reef monitoring program use sampling procedure to gather precise data on reef ecosystem, revealing species diversity, distribution, and health. These methods are methodological, allowing researchers to compare data from various locations and time period. The selection of sample procedure is based on specific objectives, such as coral cover, fish number, and water quality measure.
Hi, I am AIMUNI Hasnan. First technique is light intercept transect that used to identify colony size and percentage cover of benthic communities. The equipment is fiberglass measuring tape, underwater slate and pencil, underwater equipment for divers, and also observers and boat for driver. Procedure is set up the line transects, next move along the transect to observe and record the organisms found. The advantages of LRT is require fewer equipment and easy to set up. It most accurate to estimate percent coral cover, provide comparable data and colony size. Limitations are require high level of expert and expensive, time-consuming, limitation on monitoring objective, and more. Second technique is point intercept transect (PIT), used for determining the condition of coral reef benthic communities. The equipment are plumb line, tape measure, underwater housing, underwater card, slate and pencil, and body steam. The procedure is set up the line transect, move along the transect, and record the benthos found. The advantages are easy to learn and apply, most accurate to estimate percent coral cover, permanent record by photographies, and more. The limitation of PIT is not suitable for identifying rare species, could not able to measure rugosity of coral reef surface, and high precision of spatial variability of reef depend on the number of points required.
Assalamualaikum, I am IQBAL and now I am going to present about video transect. The video transect method involves locating organisms at random location within a rectangular region above a coral reef. Now, the equipment that are used in the video transect method, there are two of them. The first one is field equipment. Now, for the field equipment, there will be tape measures, digital video camera, underwater housing, appropriate color filter, underwater equipment, underwater camera, and also video head cleaning cassette. The lab equipment. When it is used in lab, it will require a television monitor with 5-point arranged in a face-centered cube on the screen, a video recorder, a database software for data entry, and also a personal computer. These are the procedures when applying and using the video transect method. Moving on to the benefits of using the video transect method. The first one, it can be used by experienced divers without knowledge of marine life identification. The next one, faster than still photography, provides a record that is permanent, useful in a majority of diving circumstances, including low visibility, and also enables later comparison between observers. Finally, for the last part on the video transect is the limitation. Now, the software is pricey but readily available. Analysis is expensive and time-consuming. Next one, expensive machinery that needs ongoing upkeep. Results take time since tapes need to be analyzed first, and finally needs a secure location to store video, equipment, and records. Now, moving on to the next method that will be presented by me is the visual quadrant. Now, visual quadrant is a traditional method for studying ecology, particularly biodiversity. Now, the equipment that are going to be used for this method is a 25 centimeter squares, will cut out of a 1 meter cube PVC quadrant. The next one is transect tape, a 10 meter transect line with markers every 1 meter, 3 meter, 5 meter, and 7 meter, and also 9 meter, a plastic roller, and a 25 centimeter cube quadrant. On to the advantage and limitation. The advantage for this method is that it is cost effective, less likely to overlook small, rare, or cryptic species in small quadrants, information in depth about the types and abundance of algae. Now, the limitation for this method is that it is time consuming and estimate can differ depending on the observer. We advise all observers to receive standard training for point quadrants which have lower human error rates, nonetheless have higher precision. Thank you so much. I will be passing the presentation to the next presenter.
Assalamualaikum, my name is Nurul AZMIERA HUDA binti Ibrahim, and I would like to present to all of you about macroinvertebrates belt transect. In reef monitoring, belt transect sampling is a useful technique used to thoroughly evaluate the composition, distribution, and health of marine species within a specific area of the seabed. The study of benthic species such as coral, algae, and invertebrates, as well as the characteristics of the surrounding habitats, is particularly well suited for this method. This belt transect sampling technique is described in more detail below, along with an explanation of its importance of reef research and conservation efforts. So next are an equipment that used in a belt transect as you can see in the slide, such as first measuring tape or rope, second digital underwater camera, third compass or GPS device, and appropriate closing. Other than that, a procedure that we get is first study area selection, second transect line placement, third belt width selection, fourth data collection, 5 replication, 6 data analysis, 7 interpretation, and last reporting. Next, advantage in BED Transit is Transit offer a true tool for ecological evaluation. Secondly, BED Transit's standardized sampling method guarantees data collecting uniformities. Lastly, BED Transit also give considerable spatial knowledge. Last but not least, a limitation in BED Transit. So first, to monitor coral reef using the BED Transit sampling approach. Secondly, BED Transit technique limited coverage is one of its main drawbacks. And lastly, BED Transit survey can have significant labor and time requirement in terms of logistics.
Which belt transit method is a quantitative research tool used to identify the number, size, and species composition of reef fishes within a predetermined volume. Researchers use a transit line to record information about the fish community and their distribution. First preparation, we need to determine the habitat you want to investigate. Next, choose a site section of the habitat. Third, set up the transit. Then, diving and data collections. Next, watch the different fish species that come into your area. Then take pictures or movie of the fishes species. Next, notes and environmental collection. Then transit completion. Then put the information gathered about fish species in order. Lastly, do observation and report. These are all equipment needed. The advantages: first, provide quantitative data on fish evidence and distribution. Next, researchers gain insight into the composition and dominance of various populations within habitat. Third, availability allows researchers to allocate resource effectively while still obtaining valuable ecological data. Lastly, researchers can gather data without significantly disturbing fish behavior. For the limitation, first, this limited sampling area may not fully represent the overall fish community composition. Next, strong current can be challenging for divers and may limit their feasibility. Third, microscale intersections and fine-scale habitat features might be overlooked. Lastly, variability in sampling effort can affect the accuracy of data comparison between different transit or sites.
So, a conclusion that can we get is the health and dynamic of coral reef ecosystem, which are essential to marine biodiversity and the balance of the world's ecosystem, are best understood through reef monitoring. That's all. Thank you.